All right, let's get right into it. We're going to be looking at one of the biggest, most confusing puzzles in all of modern biology. It's a mystery that's literally written into the DNA of every living thing on this planet.
The immense variation in genome size across the tree of life is one of the most remarkable and historically bewildering observations in genomics, contextualized primarily by what was known for many decades as the C-value paradox. So that's the core idea, the central puzzle that we're going to unpack. Okay, so to get a handle on this, we've got to start with the observation that first sent scientists down this rabbit hole. It all begins with what feels like a really simple, almost obvious rule. You know, when you look at the big picture, there seems to be this general trend. It just makes sense. The more complex an organism is, the more genetic instructions it should need. It's like the more complicated the building you want to construct, the bigger the blueprint, the DNA, has to be. So if you look at the very bottom of that complexity ladder, you've got viruses. I mean, these things are incredibly simple. They're basically just a little bit of genetic code wrapped in a protein shell. So it's absolutely no surprise that they have the smallest genomes of all. All right, take one step up from there, and we find prokaryotes, things like bacteria. Now, they're still single-celled organisms, but they're definitely more complex than viruses. And just like we'd expect, their genomes are bigger. So far, the rule is holding up perfectly. And then, all the way at the top, you've got the really complex multicellular life. We're talking eukaryotes like plants, animals, and of course us. It seems totally logical that we'd have the biggest, most packed genomes to build our incredibly intricate bodies. And that's the basic, intuitive rule we're starting with. But this is where it all gets really, really interesting. Because the moment you start to look a little bit closer, that simple, neat, tidy little rule, it just completely falls apart. It shatters. So let's meet our first paradox maker, the humble amoeba. I mean, look at it. It's just a single-celled blob. It doesn't have organs, it doesn't have tissues, no complex behaviors to speak of. It is pretty much as simple as a eukaryote can possibly be. But here is the absolute kicker. The amoeba's genome can have more than 10 billion base pairs. Now, think of base pairs as the individual letters in its genetic alphabet. This tiny, simple creature has a genetic instruction book with over 10 billion letters. That is a truly monstrous amount of DNA for something that seems so basic. Okay, now let's put that into perspective by comparing it to, well, us, humans. We're arguably one of the most complex life forms on the planet. We've got all these specialized organs, incredibly intricate developmental pathways, you name it. And our genome size? It's a comparatively tiny 3 billion base pairs. You heard that right. That single-celled amoeba has a genome that's more than 3 times larger than ours is. And that, right there, is the heart of the paradox. The simpler organism, the amoeba, has way, way more DNA than the more complex one, the human. It's clear proof that the amount of genetic material an organism has just does not line up with how complex it is. You know, the mystery only gets deeper. It's one thing to compare distant relatives like an amoeba and a human, but it gets even weirder when you start looking at species that are super closely related. Let's take a look at our first case study, 2 species of shovelbugs from the genus Hostorius. Now, these little guys are as closely related to each other as humans are to chimpanzees. They look almost identical, and they live in the same kind of beach environment. Okay, so the Canadian shovelbug, Hostorius canadensis, has a huge genome. It clocks in at 14 billion base pairs. To put that in perspective, that is more than 4 times the size of the entire human genome. But then you look at its close relative from Texas. Its genome is only about 3 billion base pairs, almost the same size as ours. So here's the really mind-bending part. One bug seemingly needs an extra 10 billion lines of genetic code just to build a creature that looks and acts almost exactly like its cousin. And this isn't just some weird thing with shovelbugs. This phenomenon is so common that biologists actually have a name for it. They call it the onion test. This comes from the genus Allium, which, you guessed it, includes all the different kinds of onions and their relatives. Get this, within this single group of plants, the genome sizes range from 7 billion base pairs all the way up to a staggering 31.5 billion. That's a 5-fold difference. It means one onion needs 5 times more DNA than another one just to do the basic job of, well, being an onion. And believe it or not, the examples just keep getting more and more extreme. So let's take a quick tour across the tree of life and look at some of the most mind-blowing variations in genome size ever found. The pufferfish, for instance, is famous for having this incredibly tiny, compact genome, only 500 million base pairs. Then you jump to the other end of the spectrum. The marbled lungfish holds the record in the animal kingdom with a colossal 133 billion base pairs. But the champion, the current record holder for all known life, is a small fern with a genome of 160 billion base pairs. That is over 50 times bigger than our own.
When you actually see all these numbers laid out side by side, it really drives the point home. From the tiny, efficient genome of the pufferfish to the absolutely enormous one in that fern, there is just no clear pattern connecting genome size to complexity. It's just a wild, breathtaking spectrum of diversity at the most fundamental level of biology.
So all of this evidence, all these crazy examples, they bring us to this one big, lingering problem in the world of genomics. This isn't just some weird little fact. It's a deep, fundamental challenge to how we think about what a genome is and what all that DNA is even doing. Thus, the problem is, if anyone claims that the entire genome is functional, then they have to explain why one species of onion needs 31.5 billion base pair, whereas another species of onion from same genus needs only 7 billions. They also need to explain why mucouchees need only 0.5 million base pairs, whereas the lungfish needs 133 billion base pairs.